How's everybody doing today? Good. Everybody survived one day of biochemistry. This is good news. I did too, which I'm happy with. That's good news too. So a couple of things. So first of all, I apologize. I usually have the videos done by the next class, and I haven't finished the video. I'm trying to do something a little different with video this time, and so I'm to improve it. And that's taking a little bit longer than I anticipated. So it will certainly be up by sometime tomorrow, but I can't promise anything before then. And of course, I'm already getting behind because I'll have to get today's posted as well. But I promise you I will be on top of it. The things that I'm doing to the videos, I think, will actually improve them quite a bit. And they were in pretty good shape to begin with, but I like to have them as clear and as nice as possible. One of the things that one of the few things students have complained about in my videos is that my microphone only picks up mono. And so if you listen to them, you only get it coming out of one ear on the speaker. And so people find that very annoying. So I'm having to do some tricks to make it come out as stereo, and that's one of the things I'm messing with right now. So I'm hopeful that you'll have a balanced listen to my videos once they are done, and we'll see. So I'm doing a little experiment today. Hopefully it worked on yesterday's video, and if not, we'll get it going. But I promise you'll have them. Okay? I also will have the highlights set up for you as well, and I usually do those at the same time as the videos go up. Okay? Now, I didn't give you my email address, and my email address is right over there. If you would like to see my calendar, send me an email. I'll send you a link to my calendar. I'd be more than happy to send you that. And I'm also going to post a link to that on this page as well. So if you're just looking for my calendar, it'll be up here eventually. Okay? My policy on meetings with you is I want to be as available as I possibly can. Okay? So I don't keep specific office hours. My policy is if I'm not scheduled for a meeting on my calendar, I'm in my office and available to you. If you want to schedule a meeting, I'm happy to schedule you a meeting, but basically I'm there, and so I want to be able to be available to you. Okay? That means you don't have to say, oh, I have to skip class to go see Kevin's office hours, because I have a lot of office hours. I'm there most of the time. Okay? You'll see when you look at my calendar, I have a fairly busy calendar, but nonetheless, you guys have priority. So if you come to my office and I'm talking to somebody, all you have to do is stick your head around the corner of my office and say, hey, Kevin, here I am. And I will either make an arrangement with you right there, or I will try to get you in at that point. Okay? So I want to be available and accessible. The two TAs from yesterday have also set up office hours. I will also post a link to their office hours on here. That should be pretty straightforward. They are also available to help you, and I'll have the information about where and when about that. The third thing, there's only three things. The third thing today is I'm going to finish a little bit early. I know that's going to break a lot of hearts, but I will finish a little bit early today. We don't have that much we have to do. So I have two lectures to cover some fairly important material. And it's one of the things that in this class that students get hit right in the face right up front. Okay? Oh my, these things I didn't get in my freshman chemistry. Okay? These things about pH, these things about calculations, okay? The whole class is not about pH, the whole class is not about calculations, but these are the first things you're going to see in the class, okay? So this is where anxiety comes in and so forth, and that's why I want you to come see me if you're having trouble working some of the problems that I give you and so forth. You're going to see that my approach to chemistry and my approach to math is going to be different from most people's, and that's to make it easier for you. I think that students should be taught a practical way to look at chemistry and a practical way to look at math that has very little to do with what most people see with chemistry and math. I'm going to memorize this. I'm going to punch this in the calculator, and that's the two versions or the two things that they've learned in chemistry and math. And I think those are awful things. Awful things. In my BB 450 class, I don't let students use a calculator. Oh, look, everybody's looking around the room right now. Is he going to do that to us too? Okay. And the reason I don't is because students have gotten to the point with a calculator where the calculator is everything that they want to do, but they don't even learn the math as a consequence of that. Okay? Now, I let you use a simple calculator in here, but you will discover you will not need a calculator in here. Okay? Because I'm going to teach you math and I'm going to teach you chemistry in a very practical sense that you're going to discover you're not really going to need it. And moreover, you're going to discover that that calculator will frequently lead you down a path that you don't know where you're going because of the way we've taught people math and chemistry. So bear with me. Okay? We'll go through that adventure, and if a calculator makes you comfortable, you'll have a calculator to work on the exam. All right? Now, that's the bad news. The good news is I'm not going to make you memorize any formulas. I will give you every formula you're going to need for the exam. I'll have it right there on the front page. I'm going to show you some formulas today. You're not going to memorize any of them. Okay? So that means now that you're going to be using this information in a practical sense and not wasting your brain power on memorizing something that really you could look up. All right? So I'm much more interested in seeing how you can use an equation than how you can memorize an equation. Okay? This is a practical sense. All right. Well, I'm getting ahead of myself. Let me start with some very basics about chemistry that probably will be review, but in many cases probably weren't taught to you in a practical sense. So we're thinking practical here. You're going to get tired of that word because I keep saying that word over and over and over. It's like walking, right? You lecture and you get stuck on a word, and all of a sudden, every time you open your mouth, that word comes out. Water. Water is where we start a discussion about chemistry because water is essential for life. We don't know of any living system that is independent of water. You guys all know from basic biology that our cell, that our body probably consists of about 2/3 by weight of water. We're walking bags of water. Okay? That water is encapsulated within cells. It's also carried between cells. But for the most part, we are an aqueous organism. We need to understand water in order to understand biochemistry. Water enables cells to do things, but water also provides some limitations for cells, and cells have adapted to those limitations with some of the things that we're going to see. Okay? But every living system that we know of requires water. Okay? Well, one of the things you learn in freshman chemistry, I hope, is the notion of polarity and electronegativity. Okay? Now, I'm not going to call on anybody and say, give me the definition of electronegativity. I'm going to tell you what that basically means. Electronegativity means desire for electrons. Okay? Desire for electrons. You might have a desire for a Twinkie. You might have a desire for a sandwich. You might have a desire for a Dutch Brothers Slurpee. Okay? Which looks pretty good, by the way. All right? That desire is going to make you want to have it, right? For an atom at the atomic level, that desire means I'm going to grab onto you and hold onto you tighter. Okay? Not all atoms have the same electronegativity. Oxygen has a fairly high electronegativity, and higher electronegativity means greater desire. Oxygen has a high electronegativity. I can't talk today. It has a higher electronegativity, which means it wants to hold electrons closer to itself. It's very fond of those little electrons. If I have two atoms in a bond, and one has a higher electronegativity than the other one does, the one with the higher electronegativity will tend to have the electrons closer to it than the one with the lower electronegativity. Okay? Let's look, for example, at oxygen and hydrogen. Oxygen has an electronegativity of 3.5. Hydrogen has an electronegativity of 2.1. If I have a bond between hydrogen and oxygen, that means that oxygen is going to hold the electrons closer. Hydrogen is going to have the electrons farther away. And so if we measure charge by the proximity of the electrons to the nucleus, we can say, well, oxygen is going to look more negative than hydrogen is. That means oxygen is going to look more negative, and hydrogen is going to look more positive. It's not the same as an ionic bond, like NaCl, because the difference between those electronegativities are so great that we can say, well, chlorine takes that one, and sodium gives it up. We can say it's a plus or a minus, right? when we're sharing electrons, as we're doing here, and there's just a subtle difference between them, we can only talk about what we call partial charge. So if we had water, H2O, we would look at water and we would say that the oxygen will tend to be more negative, partially negative. The hydrogens will tend to be more partially positive. Now, that's what we're going to be concerned about here, okay? Because that gives rise to everything that we know and everything that's important about water. Water is basically a molecule that is unbalanced in its charge. The oxygen is more negative, the hydrogen is more positive. You notice that the bond angle is such that this guy is bent. That's the structure of water. If I were to be looking at water from this direction, I'd say, oh, wow, look at all that negative. If I were to be looking at it from this direction, I'd say, wow, look at all that positive. Okay? Well, we know, of course, that water doesn't exist by itself. Water exists in solution when it's liquid. There's billions and trillions and godzillions of these things. And we know from our basic chemistry and our basic physics that plus attracts minus. So instead of having a single isolated water molecule as we have here, okay, oh, wrong one. All right. We have something much more like this, okay? We have a variety of molecules here. Here is something that has a negative charge. Here's some positive charge. That little delta sign there means partial. You can just assume that delta right there means partial. So partial times 2 minus, partial times 1 plus. Well, this partial 2 minus is going to be attracted to that partial 1 plus. Negatives attract positives. All right? Now, this really turns out to be absolutely critical. If this weren't true, we wouldn't be sitting here. Life on Earth would not exist if this weren't true. That's how important this is. Life on Earth would not exist if this phenomenon I've just described to you were not true. Okay, I'll show you why in a second. This phenomenon is known as hydrogen bonding. You've heard of hydrogen bonding before, right? Hydrogen bonding is something that when we have water, we always have some kind of hydrogen bonding going on because of this interaction. This hydrogen bonding might be between water molecules. It might be between water and other molecules that have hydrogen bonding. Okay? It might even be between molecules that don't even have water. And we'll see a little bit of an example of that later. All we have to have hydrogen bonding is have something that's got greater affinity for electrons than hydrogen does. Something that's got greater affinity for electrons than hydrogen does. Usually that's oxygen, but it could also be nitrogen, as we will see later. All right. Now, each one of these little bonds between, let's say, here and here, each one of these little bonds between one of these partial negatives and partial positives is not very strong. But collectively, when we have trillions and trillions and trillions of these in a glass of water, the effect is very large. Okay? That's a very important point about hydrogen bonds. Individual hydrogen bonds are very, very weak. Collectively, hydrogen bonds are very, very strong and very important. Okay, we'll see why that becomes important later when we talk about proteins. All right. We know that water and oil don't mix. Okay? You go get your salad dressing, you've got 2 layers, right? You shake it up to mix the vinegar with the oil. Vinegar is a water-based solution. And when you shake it, you see the whole thing mix up, and if you let it sit for a while, they separate. Why do they separate? Well, in this case, likes stay with likes. Okay? The likes are hydrogen-bonded molecules like each other. Things that can't form hydrogen bonds don't like each other. Or don't like things that do have hydrogen bonds. I should back up. Things that have hydrogen bonds like each other. Things that don't have hydrogen bonds don't like things that do have hydrogen bonds. So oil doesn't have any hydrogen bonds of any significance in it. Virtually no hydrogen bonds in oil. Water's full of hydrogen bonds, so water stays with water, Oil stays with oil. Okay? Most of the things that we have in our bodies form hydrogen bonds. It's pretty important because we've got 70% water, right? Proteins form hydrogen bonds. Sugars form hydrogen bonds. Amino acids form hydrogen bonds. DNA forms hydrogen bonds. RNA forms hydrogen bonds. There's a class of molecules in our body that don't form hydrogen bonds very well. We call them lipids. Lipids. Okay? We'll talk more about lipids, but a really good category of lipid is fat. Okay? Fat. Fat is so difficult to interact with water that we have specialized cells in our body that store our fat. We call them adipocytes. Okay? They're designed to sequester and hang on to fat. And one of the things we'll see in our bodies is that fat is really great for storing energy, but it's a real challenge for the body to deliver it where it's needed. It's a real challenge. We'll see that later. Okay. And it all comes down to the fact that fat doesn't form hydrogen bonds with water hardly at all. Okay. Amphiphilic. You guys have probably heard that term. What does amphiphilic mean? Okay. When we talk about hydrophilic, we talk about hydrophobic, and we talk about amphiphilic. So let me give you some definitions for those terms. Hydrophilic. The name tells us what it is. Hydro meaning water, philic meaning like. Something that's hydrophilic likes water. It will interact with water. It will dissolve in water. Hydrophilic things will include what I just described to you: sugars, nucleotides, DNA, RNA, most proteins. Okay? The opposite of hydrophilic is hydrophobic. Phobic meaning fear of water. Things that won't dissolve in water will include fat, lipids, the fat-soluble vitamins. Okay? These are hydrophobic. There's a third term that we use called amphiphilic. And amphiphilic means they have a bit of the characteristics of both. Okay? A really good example you see on the screen is called palmitic acid. Palmitic acid is an example of what we refer to as a fatty acid. Fatty acids, we will see later, are one of the building blocks of fat. But a fatty acid by itself is amphiphilic, meaning part of it doesn't like water at all. This portion over here that you see, the black with the white bonds, those blacks are carbons, the whites are hydrogens. Those guys right there, by the way, carbon and hydrogen have very close electronegativities. They share electrons very easily. One doesn't pull it one way or the other very much stronger. So there's really very little difference in the charge of a carbon-hydrogen bond. Carbon and hydrogen, carbon bonded to hydrogen, okay? Look at this end. This is a carboxylic group. Carbon double bonded to oxygen and also bonded to an OH. This guy is polar, meaning that it has an uneven distribution of charge because there's an oxygen and there's a hydrogen, and what do you suppose is the nature of that bond? It's unequal, right? An unequal bond, an unequal sharing of electrons means it's polar. Just like I showed you water, one is going to have a more negative and one is going to have a more positive. And from what I've told you, oxygen, of course, is going to be more negative. The hydrogen is going to be more positive. What we will see with respect to this group later on is that this group will actually ionize, meaning it can readily lose its hydrogen. Ionization means it readily can lose its hydrogen. Okay. Now, I said this was amphiphilic because part of it doesn't like water, meaning it's hydrophobic, and part of it does like water because things that are charged or partially charged will interact with water. So this guy has both properties. Amphiphilic molecules, you'll see, like this guy, function very well as detergents. They help to solubilize things that don't like to go into water. All right. Here's an example of that. a micelle. Okay? This is a schematic representation of what happens if I take that palmitic acid and I put it into water. This guy has two different components. The inner component here is the part that doesn't like water. You'll see that those parts are all interacting with each other. The part that does like water are these negative charges out here because they've lost that hydrogen and gained a negative charge. They love water. So this forms a nice little ball we call a micelle. Well, that's kind of cool, but more importantly, when we wash our hands, the reason this gets rid of our grease is grease doesn't like water. The grease goes in here. This carries the grease away in an aqueous solution. So we wash our hands, the grease comes off, and it's tricked into being dissolved in water. If we do the grease by itself, it won't go into water, but this guy allows it to be placed into water. So micelles come from amphiphilic molecules. By the way, I love questions, so if you have a question, raise your hand. I don't have any hesitation in answering questions. Clear as mud. Yes?
Is amphiphilic compounds or substance is important for transportations in the cell?
Oh, very good question. So her question is, are amphiphilic substances important for transportation in the cell? It's a very good question. It turns out that within a cell, the answer is basically no. But outside of cells, they turn out to be critically important. How many people have ever heard of LDLs? If I say LDL, what comes to your mind? Low-density lipoproteins. You memorize that name. But what comes to mind in terms of the phenomenon? Why do we have LDLs? Anybody know? Transport fats. Very good. One of the things people associate with LDLs is cholesterol, right? Your LDL levels are high, your doctor says that's not good because your cholesterol levels are high. Cholesterol is a lipid. Cholesterol doesn't like to be dissolved in water. Fat doesn't like to be dissolved in water. How do cholesterol and fat move in our body? By being carried in LDLs. These are amphiphilic-like complexes that carry these things in our bloodstream. So outside of our cells, amphiphilic molecules play a very important role in the body. They will also carry fat-soluble vitamins. And yes, cholesterol is very important to be carried. Cholesterol is not just a bad molecule. Very important molecule to be carried. Okay. There we go again. This is a better representation now of just water. Here's that partial positive attracted to that minus. And we can think, wow, in a 3D environment where all this water is located, that is really a very complicated structure, okay? Well, it turns out it is complicated, and a very important thing happens, okay? Here's some more interactions. Here's a hydrogen bond between, let's say, two waters. Here's a hydrogen bond between a water and a carbon-oxygen bond. We see this frequently happening inside of our cells. Here's something between an amine, which is what this NH refers to, and water. Amines are very important in proteins because that's where amino acids get their names. This helps proteins to be water-soluble. Okay? And a variety of things. We can have bonds between an amine and a carbonyl group. This happens within a protein, also within a nucleic acid, as we shall see later. So this hydrogen bonding starts popping up everywhere when we look around biologically. Here's some more examples. Water with an amine. You can see it in a different way here. Here's water with a carbonyl group. Here's water with an alcohol. Alcohol is water soluble. You guys go to happy hour, you know that, right? If you didn't know that, maybe you probably don't remember that after a few alcohols anyway, right? Okay. All right. Now, energetically, we have to think of energy. I told you, we said we're going to spend a lot of time this term thinking about energy. Energetically, we can measure the strength of a bond by how much energy it takes to break it. Stronger bonds will take more energy to break than weaker bonds will. I could measure two different ropes, for example. One very thick rope, one very thin rope. and I can see how strong each rope is by measuring how much force it takes to break the rope. Same thing is going on with these different kinds of bonds. All right? I've been talking about hydrogen bonds. Here's the amount of energy that it takes to break a hydrogen bond, and no, you don't need to know that number. Okay? This is a measure of how much energy it takes to break a hydrogen bond. Look how much energy it takes to break a covalent bond. A bond between an oxygen and a hydrogen, or a bond between a carbon and a hydrogen. A covalent bond where there is sharing of electrons is very difficult to break. Okay? Hydrogen bonds are weaker, but collectively, they're very strong. Okay? Individual hydrogen bonds are weaker, collectively, they're very strong. So if I were to ask you which is stronger, you would, I hope, tell me that a covalent bond is much stronger than a hydrogen bond is. Okay? That'll turn out to be important later. Okay. Now, I told you earlier that if we didn't have hydrogen bonding, we would not all be sitting here. And this figure right here tells us why. All right? Let's take a look at this. The molecular weight of water is 18. If everything else were equal, molecular weight should determine the boiling point of a molecule. That is, if there were no hydrogen bonding, the only thing that would determine if something was liquid or solid would be how heavy it was, how dense it was. Well, let's look at the boiling point of water and compare it to the boiling point of something that has about the same molecular weight. Ammonia has a molecular weight of 17, water has 18, methane has 16. Look at the boiling points of these. What does this mean? This means that are we going to have any liquids below this, above this temperature? No. The temperature on the face of the Earth is usually above minus 33 degrees centigrade. If water were like these guys, water wouldn't become liquid until it were in the Arctic or the Antarctic. What would happen to us? We wouldn't be here. This happens because all of those hydrogen bonds collectively are attracting and holding on to each other for dear life. And if we want to break those bonds, we have to heat it up to a certain temperature before those hydrogen bonds get broken, because breaking of hydrogen bonds causes water to go into the gaseous phase. Okay? These guys don't have hydrogen bonds. At least they don't have very significant hydrogen bonds. Methane has essentially no hydrogen bonds. Ammonia has a little bit of hydrogen bonds. Okay? Water has much more hydrogen bonds, and we can see that the hydrogen bonds are actually determining the place where they melt and also the place where they boil. So hydrogen bonds are essential for us to have life as we know it on the face of the Earth. Okay. Now, I hope I've convinced you that hydrogen bonds are important. All right? Now I want to talk about another important consideration about water. Water facilitates ionization. If I say ionization, what do I mean? It means creation of an ion. Creation of an ion means basically isolating one atom from another atom. Okay? With an uneven exchange of charge. Here's ionization. Okay? Very simple ionization. On the left, I see a schematic representation of an acid, HA. On the right, the acid has ionized. One part got the electron, the hydrogen didn't get the electron, and the hydrogen only has one electron, so when it loses its one electron, hydrogen is left as a proton with a positive charge. Okay? This guy on the left we refer to as an acid. In this class we'll refer to it as a weak acid. If you take a weak acid and you pull the proton off of it, you create a salt. That's what the A- is. Okay. People think of the proton as the acid, but the proton is not the acid. A proton is what an acid donates. Protons are not acid. Protons are what acids donate. Now, what you learned in freshman chemistry was mostly strong acids. You worked with HCl, right? Does anybody remember freshman chemistry lab? Okay. Yes, you worked with HCl. What's the difference between a strong acid and a weak acid? Oh, boy. Really important difference. Okay? And it's dramatic. If I take HCl and I put it into water, it will do completely this. Every single one of these I start with on the left will turn into one of these and one of these. Meaning if I start with 1 million HCl's, I will get 1 million protons and I will get 1 million Cl minuses. Okay? I will have 0 HAs left over. That means it completely ionizes. That's the definition of a strong acid. A strong acid is an acid that completely ionizes when you put it into water. HCl is that way. HNO3 is that way. H2SO4 is that way. Okay? That's a strong acid, and that's most of what they focus on in freshman chemistry. HCl is pretty nasty. Did you ever get HCl on your skin in freshman chemistry? You were very good students. You were careful, and they told you wear the gloves and everything, right? But if you get HCl on your skin, it burns. HCl is not good for you because it completely dissolves. It gives a lot of protons, and as we will see, donating a lot of protons is really a problem. Okay? What's a weak acid? A weak acid is something that doesn't completely ionize when we put it in water. All right? An example I will talk about in this class a lot is acetic acid. Acetic acid is the acid part of vinegar. Okay? If I take acetic acid and I dump it into water, I make vinegar. That's how we make vinegar. If I take 1 million of those acetic acids and I put it into water, I will not get 1 million of these and 1 million of these. Okay? I may get a few thousand of this and a few thousand of this, but I will not get 1 million of this and 1 million of this. So let's say for the sake of argument I get 1,000 of this, I get 1,000 of this. How much of this am I going to have? Well, 1 million minus 1,000 is going to be 999,000 of these that are going to be left. That's very different than a strong acid. Very, very different than a strong acid. Now, that's something you're going to forget, which is why I keep repeating myself. That's very different than a strong acid. That's very different than a strong acid. Everybody with me there? Weak acids do not completely dissociate when we put them in water. Okay? Now, that turns out to be very important when we start doing calculations about the pH. If I said to you, what's pH? What would you say? I know what the mathematical definition. What is pH a measure of? It's a measure of the concentration of hydrogen. It's a measure of the acidity, both right. The lower the pH, the more acidic a solution is. The higher the pH, the less acidic a solution is. Right? Everybody with me? Okay. So, if I take acetic acid and I put it into water and I spill vinegar on myself, it doesn't burn like HCl does. One of the reasons, it doesn't dissociate as much as do the protons in HCl. I can put vinegar into my salad and eat it, and it tastes really good when I mix it with the oil, but if I tried the same thing with HCl, I would die. Therefore, I don't recommend that. Make sense? Okay. Now, this ability of weak acids to not completely dissociate turns out to be really cool and really important. Okay? Now I'm going to introduce this. Tomorrow I'm going to spend the entire period talking about this in some detail. Okay? Let's imagine for the moment that I have an acid that is half dissociated. Let's say I take that 1 million of these, and I have 500,000 of these, and I have 500,000 left of these. Okay? Everybody envision that? Forget about the protons. Because the protons we're going to see can be varied very easily. We can manipulate the protons, so just because I have 500,000 of these and 500,000 of these doesn't mean I end up with 500,000 of these. I might start with 500,000, but I'm not necessarily going to end up with 500,000. Okay? That's one of the places where you're going to trip up. This and this are important for doing some calculations we're going to see. That's down here. I'll save that for tomorrow. Now, first I want to introduce why what I just told you is important. Okay? I've got 500,000 A minuses. I've got 500,000 HAs. Everybody envision that? We don't know how many protons there are there at the moment. Somebody took the protons away. Okay? So we don't know. We always have to calculate how many protons are there. Strong acids, I know how many protons are there. Weak acids, I don't know that. All right. Well, I've got 500,000 of these, I've got 500,000 of these. I'm sitting around and I say, okay, well, I'm going to dump in 10,000 protons into this solution. What's going to happen? Well, the first thing you're going to say is, well, the protons are going to go up by 10,000. And that would be true for a very micro instant. But in fact, this system that I just described to you is able to absorb those protons I just put in. Why? Because if I put too many protons in, this equation runs backwards. The A minus absorbs the protons and becomes HA. Whoa. That's kind of cool. HCl can't do that, but a weak acid can do that. Now, what's just happened? I dumped some protons in, but the protons disappeared. Most of them disappeared. I told you before how protons can disappear. Now you've just seen some protons disappear. I've dumped protons in there, and they got gobbled up. So let's say I had 500,000, I had 500,000. I dump in 10,000 protons. I'll tell you what's going to happen. 10,000 of those protons are going to be gobbled up. I'm going to lose 10,000 of this, and I'm going to gain 10,000 of this. I'm going to have 510,000 of this. I'm going to have 490,000 of this. Now, why is that important? Well, I've just resisted a change in proton concentration. The proton concentration didn't really change much when I added those protons. Whoa. The pH, therefore, is not going to change hardly at all. Very tiny change. I've just described to you a phenomenon we'll call later a buffer. A buffer is something that resists a change in pH. A buffer resists a change in pH. That's the last message I want to give to you, but there's one last thing I want to do before we finish. And I'll talk more about this tomorrow. I think we should commemorate this by singing a song. Okay? Please join me in singing very loudly this song. Biochemistry, biochemistry, I wish that I were wiser. I feel I'm in way o'er my head. I need a new advisor. My courses really shouldn't be such metabolic misery. Biochemistry, biochemistry, I wish that I were wiser. Biochemistry, biochemistry, reactions make me shiver. They're in my heart and in my lungs. They're even in my liver. I promise I would not complain if I could store them in my brain. Biochemistry, biochemistry, I wish that I were wiser. Biochemistry, biochemistry, I'm truly in a panic. Your mechanisms murder me. I should have learned organic. For all I have to memorize, I ought to win the Nobel Prize. Biochemistry, biochemistry, I wish that I were wiser. See you guys tomorrow.